So I'll give you a second to take a look at it, fix anything, and then I'm going to talk a little bit about it. So remember, your peripheral nervous system has two divisions, the sensory and the motor division. And so the flow of the arrows here are showing you that what communicates with the central nervous system. So your brain and spinal cord, you should know that that's the central nervous system. Everything else extends from that central nervous system as part of your peripheral nervous system. So what gives information to the central nervous system is the sensory division. And so these are a lot of the sensory neurons. The signals or the stimulus is from the environment. So you have sensory neurons in your hands. So when you touch something hot, that sensory neuron senses that stimulus, sends a signal to the central nervous system, which then sends another signal through your motor neuron to your muscle cells that cause your muscle cells to contract and you to pull away. So think about how fast that happens when you touch something hot and you pull away. All of that communication happened just that fast, all right? So we're talking about very high speed in the communication between your different neurons. So that's your sensory division is communicating to the central nervous system. So your sensory division is part of that peripheral nervous system outside of the brain and spinal cord, communicates with the central nervous system, your brain and spinal cord, and then from your brain and spinal cord, it sends back out specifically to the motor division, which is part of your peripheral nervous system. So these are the two parts of your peripheral nervous system. The motor division is nerve fibers that or neurons that are going to communicate either with muscles or glands. And then this motor division can be divided into two subcategories, the autonomic and the somatic. So the somatic is what I just talked about with pulling away from something hot. So it communicates with your skeletal muscles. So this is something that you can control, so it's voluntary. So voluntary and skeletal muscles, two key words there with the somatic nervous system. And so you can choose to contract your muscles to pick up your pencil, to take your notes, or whatever. The other part of the motor division is parts that you cannot choose. So this is involuntary. And I want to talk a minute about that. So there are two other kinds of muscles. There's cardiac and smooth muscle. Cardiac muscle, what does that refer to? The heart, right? So we can't control our heartbeat. We can't decide if our heart's going to beat faster or slower and so on. So this, we're going to put a little heart there. Cardiac is heart. Smooth muscle. Anybody know what smooth muscle is, or give me an example of where smooth muscle might be? Ellie?
Doesn't it make up like your small intestine?
Yep, it's found around your small intestine. We're going to say a different example. Your stomach. So smooth muscle often lines organs and so on that contract to, like, for instance, your small intestine. It contracts to kind of push food through. It's also around your esophagus to kind of push through. It's called peristalsis. So it kind of cinches around and pushes the food down, then cinches around, pushes the food down, and so on. And so we can't control that. Our stomach has muscles that churn the food and mixing it with the enzymes and so on to help with digestion. We don't control all of that. So your smooth muscle is often around organs and things like that, like the stomach and the small intestine and so on, and then also glands. So these are all involuntary. And then these involuntary muscles we can subdivide this into also two different divisions, the sympathetic and the parasympathetic. So this says, for the sympathetic, it says mobilizes body systems during activity. And then it says fight or flight. Have you guys heard of that? Can somebody raise their hand and tell me what that means? part of it, but can we put it into words?
It's like the instinct that you have when you're like introduced to like stress, whether you like run away or you stay trying to fight away.
Yeah, absolutely. And in either one of those situations with the fighting or the flight, either one of those, you need the same thing to happen in your body. You need energy for that to happen. So a source of energy is cellular respiration. So what happens is some things like your heart starts beating faster. What that does is pump blood through your body faster, which means that you get oxygen to your cells of your body faster, which then also corresponding with that increases your breathing rate, so you get oxygen and CO2 out to get to the cells of your body, so you can do cellular respiration and have energy, and so on. And so oftentimes your body will break down excess glucose that's stored in your liver cells as glycogen, so you have extra glucose, and that heart is pumping to pump that glucose through the blood to get all the cells of your body. And so that is during activity. Whereas your parasympathetic division is when it's kind of the opposite, when you're not active. And so this, it says here, conserves energy. It says promotes housekeeping functions during rest. So therefore, you're not doing a lot of activity. What happens in this fight or flight response when you have this high activity is oftentimes your body will shut down blood flow and shut down activities that are not crucial to the moment of the fighting or fleeing. All right, so what would that be? Like, for instance, blood flow and putting energy towards digesting your food. Like if you're being chased by a bear, all right, you don't need to put energy into digesting your food at that particular moment. And so therefore there's actually little muscles around the capillaries that will restrict blood flow to that area and kind of shut that off for a little bit, the digestion part. And so then when you're not in that situation, then your body will put energy towards digesting your food and things like that. So that's what it means by housekeeping functions during rest. So I use the words in the notes, rest and digest, and that's where that comes from. And so right now we're probably all in, hopefully, the parasympathetic mode. All right, so where you are in your housekeeping mode. All right, and so that's that. How'd you guys do? All right, all right, all right. So what I'd like to do is have you look next at the second thing you should pick up. Hopefully I'm going to make your day here. What I was looking and thinking last night, and I decided to make your life easier. So for your review sheet for chapter 48, I took the I can statements and I made them into questions. And this one sheet of paper, this one would have you do your review sheet for chapter 48. So the packet, that big, huge packet that you got yesterday, I'm not going to make you do to turn in, all right? To alleviate some stress, all right? I make your day, all right? So this is the thing that you have to turn in. So on Tuesday when we take the test, you have your immune system review sheet that we've been working on. That's due, and this is due, okay? Got it? All right. Now you can still, I will upload the answers to the nervous system packet if you want to use it to kind of help to study. You can, and I'll put the answers there, so that's an option, but it's not going to be something that I'm going to collect and check, okay? So therefore, makes it a little bit more doable. There's only 11 questions, although they have multiple parts, but 11 questions there, and to kind of guide your studying. One thing, if you turn to the back, chapter number 11, is the parts of the brain that I want you to know what they do. When it says brain region, I'm just going to have you cross out the brain region column, and you can just do the functions, okay? Alrighty. And then the last two things that you picked up, I'm going to use as review. And so I wasn't sure if I was going to get to it today, and at first hour I didn't. This, if you notice here, looks an awful lot like the picture I drew in the notes yesterday. We're going to use this tomorrow, and so I'm just going to give it out to you today, but just keep it to tomorrow. We're going to see if we can describe what's happening when an signal is moving down the neuron. And then also the second sheet is when it gets to the end of the neuron, what happens, and so we're going to do that tomorrow as well. So just keep these two. So this is kind of like just a review kind of thing about the two. So the plan is still, so tonight you're going to finish up. We don't have very much in our notes left. If you notice that there's like maybe a page and a half. It's not very much. The video today will be shorter. And so then tomorrow we're going to do a couple things with that and kind of pull it all together. So tonight and tomorrow we'll be done with the nervous system. Monday we're going to review, and Tuesday test over the immune system and the nervous system. All right, so that's still the plan. And so I want to talk a minute about next week because Tuesday, I'm actually not going to be here Tuesday. Our test is going to be Tuesday. I'm having surgery on Tuesday. I have a bunion on my foot. Do you guys know what that is?
Oh, my mom had the exact same thing.
Did she have the same thing?
Yeah, she had the same surgery.
Same surgery. How was it for her?
It took her a little bit to recover, but
Wait, is that what Miss Ware had?
This is, yes, Miss Ware had, Miss Ware had it done last year, and I'm going to her surgeon because she had good luck with, she had good results with that surgeon. So it's where, your bone right here, like on the side of your foot, kind of sticks out. So my foot, like here's my big toe. Here's my big toe. So normally a foot looks something like this, right? And so my foot, the bone comes out like that, all right? And I thought I could make it to, it's just affecting, and my foot kills all the time. I can't wear heels at all, and even in flat shoes, even walking in at 7 o'clock in the morning, like it already hurts during the day. Oftentimes, driving home, I take off my shoe to like dry, like it hurts. So anyway, I need to have the surgery done. So they're gonna shave off my bone and put a screw in to straighten it, because what happens is my toe actually goes in like this a little bit, and so they're going to screw it in to straighten it up and so on. So, and my surgeon only does it on surgery on Tuesdays. So that's the surgery day. So that's what's going to be going on. So next week I'm going to be would be probably on pain meds and so on and recovering. I am planning on, he told me I should be able to come back to school on the Monday after break, so that's my plan. And so it'll almost be 3 weeks then, because we have 2 full weeks for Christmas for the break. So anyway, so next week you're going to have the test on Tuesday, so I'll be here up until then to get all your questions answered and so on. And then Wednesday, Thursday, Friday, we're going to start chapter 12. We're going to go back to, so we had done 11 with cell signaling, and then we did the nervous system and immune system. We're going to go back to 12, which is all about mitosis and cell division. So just to give you a heads up of the plan, I've made one video and I'll make another video, and I'm actually going to let you watch them in class. And so it actually works out better for you probably than I'm not going to be on, because if I was here, you'd be doing it for homework probably. So so I'm going to give my sub, who you might know, she retired last year, she's got ELA, Miss Patterson. All right, Miss Patterson is my sub, and so she's going to be here Tuesday through Friday. And so I'm going to give her the links to my YouTube so that she can show you the video. So 2 days is going to be video kind of notes, and then 1 day I'm going to let you work on kind of the reading guide thing for chapter 12. Okay, so that's the plan. All right, so it'll be almost like I'm not here. So and then when we get back on after break, we're going to do a little lab with mitosis. You're going to look at some cells undergoing mitosis and different things like that. So that's kind of the plan. I also am going to have copied to give to you the review sheet for the final exam, which when we get back from break, we have 2 full weeks and then final exam week. All right, so seems like a long way away, but it's going to sneak up on us before we know it. So for those of you who like to use your time over break to, so since you have time, you can. It's totally up to you. Let me just tell you a little bit about what I do for the review sheet and the final exam. The review sheet is not mandatory that you do. Although if you do it, what I've done, if you do every single problem, every single question, and you turn it in the day of the review, I include you in a curve for the test. I give you actually a 5% curve at the final exam. All right? So what that does, it's worked. I've done this for years. It works out really well because every year I have a handful of kids that are acing the class. They feel like they don't need to do every single question on the review, so they don't do the whole review, and they're fine. But for everybody else who wants that 5%, it will be helpful for you to do the review because it'll help you to study, and you get the 5%. So that's the plan. So totally up to you. But the thing is that you can't turn it in partially done. It has to be 100. It's all or none. All right, all or none. So that's that. And it'll be due the day that we take the final exam, which is like the 25th through the 27th, I think. I looked it up last hour. So 25th through the 27th of January is the final exam. So I'll give that to you so you can have that going into break as well if you want to. Okay? Alrighty, so let's look at your nervous system tutorial. Any other questions before? Just want to do some housekeeping stuff. All right, let's go over that nervous system tutorial. All right, you guys. So let's go over this. So the first part here, you had to label the neuron. Which side of this neuron is going to receive signals? This was not a question, but which side would receive signals for this neuron? Grayson, what do you think? No, which side of this neuron, left or right, would receive the signal? The left side. So the signal that's going to stimulate this neuron would come over here and be picked up by this first line here, which is what, Tristan?
Dendrites.
The dendrites. So the dendrites, remember the signal can be a neurotransmitter that's released from another cell, or if it's a sensory neuron, it can be from the environment, like heat, light, touch, pressure, and so on. So then this line here is supposed to be showing this whole part here, which is what, Jennifer?
Nucleus?
The little green one in my thing here is the nucleus. This would be the cell body. All right, and what it's not showing you is that this is a normal cell, so it would have the mitochondria, smooth endoplasmic reticulum, all the things that a normal cell would have as well. Now, collectively, this whole thing, it doesn't have the whole, collectively, this whole part right here is called the what? This is called the axon right here. And so the axon has multiple parts. Part of the axon is exposed right here. And so when it's exposed, what do we call it? What do we call it? Yeah, the node of Ranvier. Right? And so there are lots of nodes here. And then around it is this little guy here. And this you could have a couple of different things for that would be acceptable. You could have the Schwann cell, or sometimes it's called the myelin sheath. Remember I talked about Schwann cells in the notes, that if we were to cut this in half and look at it, you'd have the axon in the middle running right through it. And then the Schwann cell wraps around the axon kind of like dough in a cinnamon roll, all right, around and around and around. And it secretes myelin, which is a fatty insulating substance. And so that's why they call it the myelin sheath, but what creates that sheath is the Schwann cell. All right, and so then the end of the axon, we have multiple ones of these, so it's pointing to this one. We call the end here, what, Olivia? The synaptic terminals, they call it. And they call it synaptic, or the terminal at the end of the axon. The reason why is because the space between, if this neuron was going to communicate with another neuron, so if I were to draw the dendrites of another neuron, that space is called the synapse. And so this little guy would release neurotransmitters to communicate with the next cell, and so that's why they call it the synaptic terminal. Alrighty, so then number 2, which structures on the diagram would receive the signal? We just sort of talked about that. That's the dendrites. And then number 3 asks you to draw an arrow to show the pathway of the nerve impulse, the direction in which it's going to travel. So it's received here at the dendrites, so you should have it going from left to right. All right, so you have that there. All right, so that's just general information about the neuron. Any questions about that? All right, so then let's look at the second part of this, which is about the resting potential of a neuron. So let me get that picture up here. So you have this picture, and it asks you for number 4, where is the greater concentration of sodium ions? And so sodium ions are in a greater concentration where, Sam? Outside. So we have our greater sodium concentration here, outside, and we have our greater potassium concentration where, Erica? Inside. And so it asks you they used term salty banana. How does the salty banana help you remember where these ions initially are?
Put some salt on the banana, you got some sodium chloride on the outside, so the sodium's on the outside. And then the banana is the potassium, so that's on the inside.
That's correct. All right, so that's a way to remember that. The sodium ions then, if the sodium ions are in high concentration on the outside as we go on in number 4, it says the sodium ions will naturally want to flow where, Lila? Into or out of the cell? Yeah, they would want to go into, so sodium will want to go from high to low concentration, which means into the cell. And thereby, potassium ions will naturally want to flow out of the cell from high to low concentration. So they're going to want to go out of the cell. And it says if a channel allows, because remember, ions need a protein channel to go through. Ions cannot get through the lipid bilayer. So these ions can only move back and forth or going from high to low concentration if you have the appropriate channels there. And so, when we look at number 5, it says embedded in the membrane are various proteins, and it asks you to list them. So there are 3 kinds of proteins. There's the sodium-potassium pump, you should have listed there, the potassium channels, and the sodium channels. And so you should have those 3. And it asks you then which of these proteins is associated with active transport. We should know this this is from our diffusion osmosis chapter, that the sodium-potassium pump is associated with active transport. And how do we know? Because it pumps things against the concentration gradient, or low to high. And every time something moves from low to high, it needs energy to go in that direction. So it pumps against the concentration gradient. So what does this protein pump do? So that's this, in my picture here, yellow here. So this is the sodium-potassium pump. So the sodium-potassium pump, it says pumps blank ions out of the cell. What kind of ions get pumped out of the cell, Elizabeth?
Wait, no, sodium.
Yeah, sodium. Sodium. And so what we have here is the sodium. The right color here. Sodium goes out of the cell, and so notice here, there's 3 here, so there's 3 sodiums that leave. And then the second part of that sentence, it says, and blank ions into the cell. And so what we have left is potassium. So potassium is going to be pumped in from low to high concentration. And these little guys here are your potassium. So there's 2 potassiums. And what this does is a couple of things. It makes it so the cell is expending energy, so you always have a high concentration of sodium on the outside and potassium on the inside. And the unequal pumping, 3 sodiums for every 2 potassiums, you're pumping more positive charges out than positive charges in. So therefore, this is also contributing to the inside of the cell being more negative than the outside of the cell. And so that's a big reason why that there's a charge difference is that sodium-potassium pump. Another reason why there's a charge difference is the unequal numbers of your potassium and sodium channels. Which one is there more of? Potassium or sodium channels? In our picture here. The dark purple is potassium, light purple is sodium, so potassium. So all of these right here are their potassium pumps. So look here. So in this case here, there's 4 of them in this example. So for every 4 potassiums diffusing out, because this is diffusion, because going from high to low, flowing through the channels, there's only 1 sodium going in. And so there's the second reason why the inside of the cell is more negative than the outside is because you have way more positives diffusing out than you do sodiums diffusing in. And it all has to do with the number of proteins that are in the membrane. There was one other reason in our notes that I talked about why the inside of the cell was more negative than the outside of the cell. Anybody remember what that reason is? Again? Yeah, there are negative ions that are stuck inside the cell. They're trapped there. Why would they be trapped? What would these ions need? What would have to be true about the membrane in order for them to get out? They'd have to have the right protein channels, right? So therefore, if the cell does not have those protein channels, those ions, that's why the ions are stuck, and that also contributes to the inside of the cell being more negative than the outside of the cell. Okay. So if we turn the page over and look at number 6, it says, notice the charge on the inside of the cell is more negative than the outside of the cell. The initial charge differentiation sets up a voltage across the membrane. That just means a charge difference. We say the value of the cell's resting membrane potential is, and we put a number value on it. What is that number, Scott?
-7.
Yeah, -70 millivolts. So the negative means the inside of the cell is more negative compared to the outside of the cell. So it's a comparison. And it asks you to propose an explanation for why there's an uneven distribution of charge across the membranes. And those are the 3 things that I just went over. So because of the sodium-potassium pump, pumping unequal numbers of positive charges across the membrane. And the second is that there's more potassium channels than there are sodium, so there's more positives leaving than re-entering, which contributes to the negativity. And the third thing is that there are those negative ions stuck as well, all right, in there. All right, and so this is called the resting potential. So inside a cell, so as we look at the next kind of section of questions, I'm going to draw my big neuron up here like I did in the note yesterday, just to kind of relate the notes to this packet here. So this is my neuron. Alrighty. Alright, so there's my neuron. Here, these are my dendrites. This is my cell body. So inside the cell is -70 millivolts. Why? Because in the membrane of the cell, you've got the sodium-potassium pump, you've got all your potassium channels and your sodium channels and so on. They're even in the membrane right here, and therefore causing the inside of the cell to have that negative 70 millivolts resting potential. All right, it's called a resting potential because this neuron is not being stimulated. It hasn't received a stimulus like a neurotransmitter or anything like that. So this is at rest, what a normal neuron is like. All right, so let's look at. Let me pull up the next picture here. So we're going to look at what happens with the stimulus and what happens with the neuron. All right, so for number 1 in part 3 here, a stimulus is applied. So number 1 says, what changes occur as the dendrite plasma membrane is disturbed? What changes? So here's the dendrites right here. What it means is disturbed is that when it receives a stimulus. So when it receives a stimulus, either from the environment, like touch or something like that. So here's your stimulus. Gets to the neuron. What happens at the dendrites? There's something in the membrane. Something happens. Something opens. Erica?
Sodium channels.
Yeah, sodium channels open. And so you have, in the dendrites, you have these sodium channels that are shut, they're gated, and they're shut. So right now, remember in the last picture we talked about how there's a high sodium concentration on the outside of the cell. And so sodium will want to move in. This sodium channel is gated and it's shut. That sodium channel only opens when a stimulus is applied to this gate. So let's say the stimulus is a neurotransmitter from another neuron. It's a chemical molecule that's released from another neuron. So what happens is that neurotransmitter binds to this. So this is your neurotransmitter. Binds to the sodium channel and triggers, that binding triggers that gate to open. And if that gate opens, so for number 1, for that first part, what changes occur as the dendrite plasma membrane is disturbed is that you should have that the sodium channels open in the dendrites. All right, so the sodium channels open. Then it says describe what will happen to the charge on the inside of the membrane near this protein channel. When the sodium gate opens, where is sodium going to go? Into, right, the cell, because from high to low concentration. So your sodium goes in, and what's going to happen to the inside of the cell now? It's going to become more positive, because sodiums have a positive charge. So what that's doing now is making your membrane potential, more positive. So it's going to become less negative. So it'll become like negative 50 now, okay? And so on. And so the positives come in. So that's what the second part of that is. It becomes more positive. And when this happens, the fancy name for that is depolarization. So we would say that right now the cell is being depolarized. It's becoming more positive. All right. So it asks you then to show the changes in charge on the diagram below. So you could pick a dendrite. So the dendrites switched around here, so it's not left to right here. So the dendrites are over here, so the stimulus would be here. So you'd have sodium gates open here, sodium comes in, and the inside of the cell will become positive. And so that's as a result of the stimulus. Now what's going to happen is, as we get into question 2 and 3, this stimulus, when the inside of the cell becomes positive, what that's going to do is it usually triggers more sodium gates to open and more sodium ions to come in, and the cell becomes more and more positive, and that those ions diffuse through the cell body and are detected by gates in the first node of Ranvier. So let's say this is the first node and this is the Schwann cell here like this. All right, so here's our, so what happens in this first node, the sodium gate is shut. This positive charge diffuses, and these gates right here at the nodes are what are called voltage-gated ion channels, meaning that they open and close as a result of a change in charge. So that's what's happening here. As these sodiums come in, the charge is becoming more and more positive inside the cell. That triggers sodium gates to open here. They're voltage gated, the change in charge, and sodium is going to diffuse in at the first node. All right, so now we'd say that this part of the cell is becoming depolarized. So when we look at our number 2, it says this depolarization in the membrane, so as it becomes depolarized, results in something called what? What did you put for number 2? Isabel, what did you put? It's called an action potential. So we're going to look here. This is the beginning of an action potential at this particular node. So it says that for number 3, the depolarization in one area of the neuron. So it says here where this is becoming depolarized for the sodium gates to open. Do you guys remember from the notes what that causes to happen over here, the next node? The next node, there are also sodium channels. There are sodium channels at every node. So there's sodium channels here that are shut. When this gate opens as a result of the stimulus, the sodium coming in here, that charge difference triggers this gate to open. Sodium comes in. This change in charge eventually is detected by the next gate, causing the next gate to open, and sodium comes in. And now this node, at this part of the cell, is becoming depolarized or more positive. So therefore, for number 3, when it asks the depolarization in one area of the neuron may stimulate other sodium channels downstream to open, allowing for the flow of sodium ions into the membrane of the axon, all right, through the membrane. All right, that's the purpose. So this node has already done its job, is to send the signal to the next node. So now what we need to do is this node, since it's done its job, we need to put it back to normal. So we need to make it back to negative. And so that's what we're going to look at if we look at the top of the page, next page. That's called repolarization. So we're going to put it back to the negative 70 here. That involves what other kind of gate that's in the node? Potassium. So potassium here, normally, so I'm going to put potassium gates here and all of these nodes here. Normally they're shut. And so what happens here is that that positive charge reaches this gate and opens and sodium comes in. Let me put the sodium here. Kind of simultaneously that's happening, this is going to repolarize over here and go back to negative. So how does it do that? The potassium gates open. And where was the high concentration of potassium, inside or outside the cell? Inside, right? So potassium is the high concentration inside, so it's going to diffuse now out. So you're going to have positives leaving. While that happens, what happens to the sodium gate? Remember, we want to make the inside of the cell negative. So how do we make it negative? Positives leaving, and then what's going to have to happen to this one? It shuts, all right? So this one shuts off. And so do you see that if sodium can't come in anymore, and now you have positives leaving, the inside of the cell is going to go back to negative. All right, so it goes back to negative, and it repolarizes. That's what it's called, going back to what it normally was. All right, so that's repolarization. So then, just so that you see this, how it happens here, while that's going on here, the sodium is coming in here. Can you tell me what's going to happen next? Sodium comes in here. What is this going to trigger to happen over here? The next sodium gate is going to open, and the next node. So now this sodium gate opens. Inside the cell becomes positive here. What's going to happen over here? Sodium gate shuts. Potassium gate opens. Potassium does what? Goes out. Inside the cell becomes negative again, and then that's repolarized. Okay? And then so it keeps on doing that at each node. All right? And so that's the repolarization part. So if we look at number 4, asks you behind the action potential, Changes occur to repolarize the membrane. Describe these changes, include what changes happen to the sodium channels and the potassium channels. So what we just did here, so what in the repolarization process, you should have that the sodium channels close, potassium channels open, and potassium diffuses out, therefore making the inside of the cell more negative. And that's repolarization. All right? And so that continues to go down the line. And then it asks you for number 5, for hyperpolarization, describe where this is and what limitations the neuron has at this time. So what happens is for this, is when the potassiums are leaving and it becomes repolarized, what happens is the potassium gates are kind of slow to shut, so what you get is more potassium is leaving than should, and so the inside of the cell actually dips below -70, so it would be like -80 millivolts. And so that part is called hyperpolarization. It's polarized more than it should be, that's what hyper means. At that particular time, a new stimulus can't trigger the, can't stimulate the neuron. And so what puts it back to normal is the sodium-potassium pump. So remember that these guys here, your pumps are in there, and it'll pump the sodiums back out, the potassium's in, and reestablish that membrane potential. All right, we're gonna run out of time here. Let me just go over, let me read off the answers to the questions here, the 5-question quiz. So number 1, you should have C. 2, you should have B. 3, C. 4A. And 5A. All right? And so tomorrow, we're going to run out of time here today. Tomorrow, if you got any of those wrong, I'll take any questions on those. We'll go over the rest of this and kind of do some review stuff, and then Monday is total review testing. Okay?